Hello all. In the unit Genetics, let us look into the topic DNA repair mechanisms. In the previous classes, you've already studied about different DNA repair mechanisms, and here we are going to continue with the remaining concepts of DNA repair. First of all, let us look into the concept mismatch repair. So where does this occur and how is it repaired? A mismatch repair may occur in a newly replicated DNA. So, when a newly replicated DNA is formed, there may be chances of the wrong nucleotides pairing. That is, thymine will always pair with adenine and cytosine will always base pair with guanine. But here, thymine may base pair with cytosine or adenine may base pair with guanine. So, this is the wrong nucleotide which has been paired. So, how is this repaired? There are special enzymes which have been synthesized, and these enzymes they cut the mismatched DNA and they remove this and replace it with appropriate base pairs. That is, they will incorporate the correct base pairs in the sequence. So there are some of the enzymes which are important in this mismatch repair mechanism. They are nucleases, DNA polymerase and ligases. The nucleases are the ones which cut the DNA and the DNA polymerases are the ones which will replace the DNA, and the gap is sealed with the ligases. This is the parent DNA strand which when replicates synthesizes the new strands, and here you can notice that the adenine has base paired with guanine. So this is the mismatched nucleotides. So in the next round of replication, this will be repaired by the previously told mechanism, that is by the enzymes, specific enzymes, and they synthesize the rectified DNA or the corrected DNA, that is with the specific base pairing. The next one is post-replicative recombination repair mechanism. This is seen usually in the double-stranded DNA. It is a double strand break repair mechanism. So here there will be a break in the double stranded DNA. So when does this occur? If there is any lesion or damage in the DNA, then this will prevent the base pairing of the nucleotides. So when there is no base pairing, there is a gap which is created in the daughter strands, and this is occurring during the replication process. So this causes break in both the strands of DNA, and this can be repaired, that is the gap can be filled by copying the sequence information from the parent strand of sister chromosomes, and this occurs by homologous recombination process, and the enzyme which is involved in this repair mechanism is DNA polymerase 1. This is the double-stranded DNA, which is the parent strand, which is depicted in black lines, and the newly synthesized strands are marked in red. During replication, there is the formation of the newly synthesized strands, and here you can visualize that there is a gap which is formed, and this gap is being repaired by undergoing homologous replication. So this gap will be filled up and it synthesizes the completely filled strands which have no errors. The next one is SOS repair mechanism. This kind of response is seen when there is damage to the DNA or when the replication of DNA stops, and also it is seen when single-stranded DNA gets accumulated. The damage of the DNA is caused by agents such as UV radiations and the chemicals such as methyl methane sulfonate. This will cause damage to the DNA and thereby it induces the SOS response. Here we have to concentrate on two main proteins. They are REC-A proteins and LEX-A proteins. The SOS response is initiated when there is severe DNA damage, and the operon operates when there is LEX A, which is a repressor of SOS operon, and the presence of REC A proteins, that is the inducer of SOS operon. So, whenever there is extensive or severe DNA damage, the replication fork will stop. That is, it does not undergo further replication, and it may initiate photoreactivation or removal of the damaged DNA. But because of extensive or severe damage to DNA, it has to bypass this process and it initiates SOS response. This SOS response in turn activates the DNA translation synthesis, or it may undergo nucleotide excision repair, and the enzyme which is used here is DNA polymerase 5. This is the double-stranded DNA which has a promoter and the operator, and there are some of these structural genes. When the LEX A protein, which is a repressor, when it is bound to the operator, there is no replication. The REC A protein gets activated and it will initiate autocatalytic cleavage of LEX A. Thereby, there is destruction of this structure of LEX A. Now, the RNA polymerase is free to move and it undergoes transcription and translation. Further, it may induce the nucleotide excision repair or the translation DNA synthesis by the DNA polymerase 5 enzyme. This is all about the DNA repair mechanisms. Thank you.